Hello, everybody. My name is Aiah Nour. I am a senior biochemistry major at the University of Nebraska-Lincoln, and my research is on determining the role of O-fucose modifications on the stabilin-2 receptor expression and function. So just giving a brief background information on our enzyme and our substrate of interest. Our enzyme of interest is protein O-fucosyltransferase 1, or POFUT1, and POFUT1 is a fucosyltransferase that hypothetically modifies over 100 human proteins contingent on a specific consensus sequence that is available on the substrate at hand. So POFUT1, in research done in 2005, is proven to be involved in O-fucosylation of a subset of receptors known as Notch receptors. And Notch receptors are increasingly important in neurological and embryo development. And any sort of lack of O-fucosylation or deficiency of O-fucosylation of Notch receptors is proven to have extreme neurological deficiencies in the embryo, and in some instances can even cause spontaneous abortions. So this increased importance of protein O-fucosyltransferase 1 modification of Notch receptors brings to question whether or not POFUT1 can modify our substrate of interest based off of the same consensus sequence. So our substrate of interest is stabilin-2, and stabilin-2 is a class H, type H class scavenger receptor. It's known as the hyaluronic acid receptor for endocytosis, or abbreviated HARE, H-A-R-E. It's also involved in, it's a phosphatidylserine receptor, and it's involved in extracellular matrix cleaning, cell fusion, and apoptic cell clearance. In addition, it's also, it regulates coagulation factors such as von Willebrand factor and factor VIII. Stabilin-2 has very high expression in the lymph nodes, in the liver, and spleen. And so our overarching hypothesis or our overarching research question is, does POFUT1 recognize stabilin-2 and modify it according to the consensus sequence like we've seen in the past with Notch receptors? So getting into the preliminary studies or the results, a research done in 2005 has identified mutants of mammalian cells, specifically Chinese hamster ovary cells, by a change in glycosylation pathway. And so we can see Lec1 here is one mutant and Lec13 is another mutant. For Lec1 mutant, we can see that it lacks the GLICNAC transferase 1, and then for the Lec13 mutant, there's a deficiency in the GDP mannose 4,6 dehydratase. And so these deficiencies and these mutants of these specific cells allows the cells to undergo alternative fucosylation pathways. And so what we did in the lab was, we can see here that we have 190 here and 315 here. Our wild type obviously shows high expression. The Lec1 cells is what we want to look out in figure 2, and we can see that there's faint expression in Lec1. This is an audio radiography analysis of stabilin-2, but we can see in the 315 hair there isn't as much expression, and so we're really focusing on the 315 hair cells because we can confidently say that the 190 hair cells are O-fucosylated. So we're really focused on the 315 hair portion, which brings us into our expression and function tests. So going into our expression test, we took 2 cell lines, a control and POFUT1 knockout cells, and we exposed them to the cDNA of stabilin-2, and then did a Western blot analysis to see expression on the 2 cell lines. And so we can see here we have extreme protein differentiation, 315 hair and 190 hair, but we can see overall that there is not really any sort of differentiation in expression, right? We see very high expression in both of the cell lines, which indicates that stabilin-2 expression is not reliant on O-fucosylation whatsoever, because we see high expression in both cell lines. Which brings us to our final figure 4, and here we test the basic functionality of the stabilin-2 receptor, its ability to clear hyaluronic acid from the cell. Its main functionality is known as HARE. And so what we did was we took 1 microgram per milliliter of hyaluronic acid and introduced it to the same cell lines that we talked about before. And we can see that these results, why they are significant, are negligible. Chief investigator on O-fucosylation, Dr. Bob Halterwinger, also concluded that these results are negligible and not necessarily indicative of any difference in O-fucosylation functionality. So just by all of these figures and by the experiments we did, we can conclude that preliminary results indicate that stabilin 2 expression and function is not necessarily reliant on POFOT1 presence. We see adequate expression and we see negligible results with the functionality test. And so the next steps is to go in and really dive into the individual epidermal growth factor domains to understand the basic molecular O-fucosylation mechanism and see whether or not the EGF domains are O-fucosylated or not. Thank you.